more time for joining the participants.
Okay, sir.
Okay, ma. Okay. Actually, how many participants, ma? Hello.
50, sir.
Okay, ma. Nice. It will take 1 hour 40 minutes, and then we'll go for a questionnaire session, or 1 hour for 45 minutes, and then go for a questionnaire session, ma.
Yeah,
Okay.
yes, sir. You can.
Okay. Thank you.
Sir, am I audible, sir?
Yes, ma. Yes, ma. You are audible.
You can start the session, sir. Participants will be joining, sir.
Okay, ma. Thank you, ma.
Thank you, sir.
Okay. My slide is fully visible now.
Yes, sir. It's visible, sir.
Thank you very much for your nice introduction, and also thank biomedical HOD ma'am for giving me this wonderful opportunity to share my research knowledge to our beloved participants and eminent professors and faculties. Okay. So today topic is clinical trials and translational biomedical research. So myself is already introduced. I am Dr. S. Sendil Kumar. I did my MTech in Anna University and PhD in Anna University, PDF in Shanghai Jiotang University, China. So I did my PDF there in a 3D cell culture model. 2021 itself, There is well versed in technology. China is well versed in technology. I worked in 3D cell culture model developments. I used hepatocyte carcinoma 3D cell culture model, organoid model development, and then after completion of my PDF, I joined as a assistant professor in Monocular Vinayagar Engineering College, and then I shifted my job to Savida University and Tech University, and right now I'm working as an associate professor in Arupadi Institute of Technology. So, so my slide is changing, ma? Excuse me.
Yes, sir. You
Okay.
have the first slide only, sir.
So, what is mean by translational research? What is mean by translational research? Actually, already you know about nowadays. Everyone talk about all the department coming together to produce a lot of human well-being products and products-oriented development, especially in biomedical applications. Now AI is booming. This translational research means collaboration among basic scientists, clinicians, policymaker, and industrial partners. Discovery from the laboratory to patient care and community benefits. Already you know about we have a lot of Siddha and Ayurveda medicines. Till now, we are struggling to implement a lot of Siddha and Ayurveda medicines. If all together and clinically proven our Siddha and Ayurveda medicine means, we can easily reach out the particular market, especially Indian product is go for worldwide like that. And also you need to prove that our Indian Siddha and Ayurveda medicine is safe and safe for everyone usage like that. Then also very important ethical reviewers and patent pathways. So you need to first identify these are the Siddha product available in Indian forest. For example, every Himalayas have different type of plant sources. Our East Coast and West Coast, everything having a different type of plant sources. You need to identify this type of plant sources. How what are the medicinal values available in that plant sources? You need to right pathway and ethical clearance to prove that this a Siddha product or this product having this much effect like that. To bridge the gap between research and clinical impact, particularly in life-threatening diseases. For example, in our in our Tamil Nadu, the COVID-19 time and all, we believe that especially the Nilavembu extract is helping to cure that particular dengue and particular COVID time and all. They are giving another one, Kabasura Kudinir. This one is helping to cure this type of diseases, virus diseases, but still now they didn't prove of clinically they didn't prove. So it is very essential. You need to prove the your products clinically. Okay, then you need lot of workforce development is available in our multidisciplinary research and trainings. For example, you need to learn from NPTEL, AICT, ATL, DST, ICMR, DBT, MSME, like that. You need to go for a variety of funding agencies and central government institutions helping towards that our product to go and reach the particular patients and particular markets. That's very important, worldwide market. Then what is meant by clinical trials? Clinical trial is the research study conducted in human to test the safety and the effectiveness of new medical, surgical, and behavioral interventions such as drug, device, or producer. Anything you need to first you discover means you need to go for clinical trials.
Sir,
So excuse me.
sir, your slide is not changing, sir.
Why ma?
Only
Clinical only
first slide is visible, sir. Okay, sir.
now
Okay,
it's changing. Yeah.
yes, sir.
So my network is problem, I think so. So
Okay.
now you can go. I will go for full mode.
Okay, sir.
Now it's changing.
Second slide, sir. It's in the second slide.
Now?
Still now it's in second slide only, sir.
Okay. So the clinical trial is very, very important. So you need to identify any type of products, anything you extracted, anything you patent righted, anything means you go for that particular medicine or drug or surgical or prototypes or device. You need to go for particular clinical trials. The worldwide lot of schools are available. Harvard Medical School, USA, University of Oxford, UK. Our aims. Um, JIPMER, CMC Vellore, NEIMER, IGAB, and several campuses are doing different type of clinical trials. Okay. So, objective of clinical trials: evaluation of safety and efficacy of new drugs, and establishing optimal doses and protocols. Once you enter in the clinical trial, means already you proven this much dose. For example, you're going for paracetamol. Okay, before and all, how much milligram of paracetamol you are taken? Nearly 250 milligram. Initially they give one. Now they are giving dolo 650. How they are increasing the dose level via this type of clinical trials? 252 now 650. Dolo 650 mg they are giving for one adult person. So each and every step they evaluated this much level of dose is essential for human being. It's have that doesn't have any side effects in that particular dose level. Like that, you need to optimal the dose and protocol, assessing the side effect and long term outcomes. You need to assess the side effect. That's why the clinical trial is important. These three different phases are involving the clinical trials. For phases, phase 1 safety and doses, then phase 2 efficacy and side effect, then phase 3 comparison with the standard treatments, and phase 4 post marketing surveillance. Once you are completed this 3 step, next you need to give the particular drugs and particular product to the uh, market, especially our consumers, our human beings or consumers. Then you need to analyze either this particular product or drug may be introduced in the market, it's have any side effects. For example, for marketing strategy and all, nowadays they are giving a sample pack to the customers. And initially they are giving the sample pack and introduce that one. For Oreo biscuit and all, they give two like that, and then finally they will introduce a full packet in with some increased price money. Like that, they will give drugs also. They will give batch wise, which batch is particularly uh, taken from people's. Is there any side effect or not? That post marketing surveillance is also very, very important. These are the phases of the clinical trial. So you can see phase 1 safety and evaluation. Safety 20 to 100 healthy volunteers needed. And phase 2 you can go for test efficacy in 100 to 300 patients with the conditions. In phase 3 you need to go for compare with the standard treatment in 1000 to 3000 patients. And for phase 4 you can go for monitor the safety in the drives. Example, you can give the all the completed all the phase 3 trials, and then it will be introduced into the market. Then post marketing survey is very, very, very essential. This is the 4 steps of clinical trial. Then you go for translational biomedical research. uh, I think you people are biomedical people, so I need to give how the translational research involving in this type of clinical trials, how it will happening. Now TBR is bridge basic laboratory discoveries and patient focused clinical applications. That is the main role of translational biomedical research. That's why our government is introduced biomedical engineering. Before and all, not there. Only biotechnology is available. Now they are introducing 2017 itself. They are introducing biomedical engineering. So it's very, very important. Translational biomedical research is very, very important. It's focusing on patient focused clinical application, aiming to improve the diagnostic therapies and health outcomes. It is a multidisciplinary. For example, fast track new innovations from bench to bedside. So you have any new innovation. For example, they are introducing the smartwatch. The smartwatch is nowadays there identifying the ECG also and your heart rate and also your how many steps are daily you are doing, walking, health monitoring, lot of pressure, O2 level also monitoring. This type of innovation is very, very important. Bench to bedside. That's a very, very important role for translational biomedical research. Excuse me. Sorry. It is a multidisciplinary. Already you know about basic research is pilot study. Anything first initially your lab study is completed, then go for pilot study, then go for clinical trials, then biomarker validation and community health interventions, ultimately impacting the health policy and practices. Why I am giving this one uh, different type of quantum dots. You can see the nanomaterial also playing the important role. It's 2.5 to 4 nanometer itself, giving different color variations. Especially it is used in bilirubin. This one is used as the biomarker for bilirubin blood sample analysis, bilirubin level identification. It's very, very helping this quantum dot also. Okay, it's anything, any materials, anything you produce, what is the impact in our society? And then you need to analyze it via this translational biomedical. So this is the bench to bedside market. You need to go for basic laboratory research, beside to community health, then applications. This is the especially bench to bedside. You need to analyze the basic laboratory techniques.
Sir?
And excuse me.
Your slide is not visible, sir. It did not get changed, sir. It is still in the second page. Yes, sir. Okay, sir. Now it got changed.
So I will go for this one. I think I will go like this, then no problem. It's visible now.
Yes, sir.
So already talking about translational biomedical research. Then how the bench to bedside, that is very, very important. So you need to analyze the patients. Then you identify how to translate our technology to bench to bedside. The basic laboratory research, bedside to community and community health. That is the main application for translational biomedical research. For example of devices, you can already I explained that one. Smart watches, smart patches, biosensor. Like that, you need to produce a different type of devices, biomedical devices. You can see nowadays you can use smart watch, smart rings, smart wallet, smart key. Like that, you need to develop different type of biosensors. The glucose monitoring biosensors, the ECG, hydration delivery medications, AI enabled smart clothing for posture, breathing, and muscle movement tracking. You can see that smart jacket. Sometimes, still now the DRDO. Yesterday I got one call from DRDO. The DRDO expert come and talk in our institution. They are giving to you need to develop one jacket for our army persons. It will be withstand in extreme cold condition and also extreme cold conditions. You need to develop. Sorry. You need to develop that type of material from via the material science. Either you can see the different type of materials in front. In the rolling, it's possible. That is the carbon nanotubes, nano walls like that. Need to develop the different type of jacket like that. They are asking. DRDO is asking to develop the low cost material like for clothing our army person. So you need to develop like the devices. Our government also giving the funding also in the type of device making. Okay. Then you need to go for focusing on 3D bioprinting and regenerative medicines. So nowadays everything is 3D printing is already you know about 3D printed statues, 3D printing everything is come. If you want your image as a 3D printed copy, it's nowadays available like that. Everywhere they are thinking about 3D printing and 4D printing and how it will be involving in regenerative medicines. For discovery, the bioprinting and patient specific tissues and organ using the biomaterial and stem cells. You can nowadays the bioprinting is also very important. The one side is 3D printing and 4D printing. They are using the material science side. Either mechanical, civil engineering people are doing. And also in our biomedical side, they are thinking about how to develop our kidney, pancreas, heart, lungs, like that. For example, they are using specific bioprinting. Especially, you need to develop the particular organ. That is the feature. Already, you know about the artificial blood is available. In Japan is invented the artificial blood. So nowadays they are trying to create your artificial kidney, artificial visions, artificial liver, like that. So bioprinted mini livers and pancreas are also important. Researchers have visualized miniature human livers, bionic pancreas constructed from patient's stem cells. You need to bring from your specific medicine is available nowadays. So you need to bring your stem cells and then go for your patient derived stem cells for this type of 3D bioprinting technology. So the period to the bio ink, they are giving the bio ink showcasing functional organoid with a vascular network. That is important in regenerative medicine discovery. Then how the fabrication will happens in that 3D printing, especially blood vessels. 3D bioprinted tissue constructed with the multi-cell types. You need to construct the multi-cell types because our human being having endothelial, epithelial, cardiovascular system, skin, everything is different cell types. Okay, so you need to multiple cell type built in the blood vessels, stimulating the complexity of solid organs and their micro architectures. There are after mapped from patient image data also. So in particular area, how to you are creating the particular, for example, you're going for some bone fracture, you need to restructure that bone fracture in that particular area. You need to scan everything and then get the CD, CD or MRI scan image, and then go for 3D printing using the bioprinter. That it will be mimicking the particular bone structure, exactly mimicking the particular bone structure. Then lungs also mimicking air sacs. The lung have some small alveoli. That particular alveoli having mimicking that particular air sacs. That is also important. They are using the visualizing of hydrogel models representing the lung alveoli, showing vascular channel for air and blood measured via advanced bioprinting systems. That is also possible. Small, you need to go for a grapes-like structure in that end of our lungs. That structure is also it's possible to create via this 3D printing. Then how this type of 3D printing model is working in a patient-specific organ model creation, or you can go for the uniform cubic or tubular structure loaded with living stem cells visualized with fluorescent dyes. So once you created all the 3D models, then how it is possible to visualize any error happens in that inside that particular organ. So they are using some visualized fluorescent dyes on that particular 3D printed. Then you can analyze that inject that fluorescent dyes. Any flaw is there in that particular 3D printed scaffold or construction. You need to analyze that one also. So then microscope to access the cell viability. How many cells are viable in that particular structure? You need to develop the 3D cell culture model. First, you need one scaffold or construction. Then you need to print your particular organ or particular structure in that scaffold. Then you need to analyze the structure having all the cell lines, cells, all the cell layers. Everything is perfectly manner is attached in that particular scaffold or not. Then how much cells are viable? how much proliferation happens, it will be growing or not, and tissue regeneration happens or not. Techniques such as cell painting are used to show the cell differentiation within the bioprinted organs. Okay, then you can go for how it will happening. You can go for step 1 imaging. You need to examine the particular patient's area, then you go for CT scan and MRI scan. Then steps 2 design approach. You need to first get it from the, I'm telling about any bone is bone or teeth. You can imagine our skull. Your skull have some damage. Your bones have some damage. Your hand bones are also have some damages. So you need to go for X-ray and then CT and MRI scan. Then second step, you need to design the particular structure. Your phone, actually, you met someone is met in an accident. This phone is fully fractured in that particular hand side. What happens? You need to rectify that issue. What happening inside? How much fracture is happening? Exactly, they are designing the particular area. That is biomimicking via using the X-ray or CT or MRI scan. Then you need to assemble the assembly using the softwares. Nowadays, Discovery 360, each and every 3D printers model having, especially uh, this one, mechanical engineering department having different type of 3D printers and 4D printers. This biomedical engineering sites also having different type of bioprinting machines. Especially in our Chennai itself, they are using this type of 3D printing for dental implants. So you can analyze the particular area of you don't have any teeth in your mouth. They are analyzing and scanning the particular area and then transfer the data to the designing approach, and then it's go for the material selections. Which material you need to go for? Here they are zirconia and paste, like teeth like paste they will using for the teeth repair and all. So you going for some other purpose for organoid development or some other purpose means you need to synthesize which type of material is required. Synthetic polymer or natural polymer or ECM, extracellular matrix needed for that. Second step, third step, material selection. Then go for cell selection. Which type of cell needed? Skin like structure, endothelial, epithelial, fibroblast cell line, cardiovascular tissues like that. You need to identify which type of cell you need to grow on that particular material selection. Then you go selected the cell selection. Then how particularly this particular cells is differentiation in that particular material selection. Like that you need to study. Then go for proliferant stem cells and multipotent. Then the generated proliferant and multipotent stem cells. Then it's go for bioprinting technology. They will prepare the bio ink, then micro extrusion, then laser assisted the particular printing will happens. Finally, it will go for the applications. These are all the main steps involved in 3D bioprinting technology. Nowadays, in future and all, you have any damages in your human organ, simply they will synthesize it via this type. Your derived stem cells automatically they synthesize your particular organ and replace it. It's possible in we are nearing that one. Nowadays they are synthesized pancreas, and also they are in China and all, they are reverse the particular type 1 type 2 diabetics using the stem cell technology. They reverse the beta cell secretions. Automatically, the beta cells will secreting insulin. Like that, the stem cell technology and this type of 3D bioprinting also very, very important to go for our organ transplantation. Okay, the future is very 10 to 20 years, people will achieving this type of organ developments. Okay, then. So designing strategy, for already told na shape and size of the particular bone, ear or skin, how much micrometer needed. You need to you have the big wound, the gap between the 2 to 3 mm or 5 to 6 mm, like that means you need personally you identify the what is the size of the wound. Then go for the which type of organ you needed, either bone, ear, or skin or not. How much resolution? Then a 3D printed tissues construction also happening. Already they are constructed in the ears. You can see the ear here. The hollow structures. You need to synthesize this type of hollow structure also possible. You can see the 3D printed tissue constructed with using different type of material and tissue organizations. You need muscle, skeletal muscle or cardiac muscle needed. They are synthesizing this type of muscles. Composite tissues. Nowadays, they are thinking about bone catalysis. Already they prepared in China and all, they prepare hydrogel for this bone replacement, bone regrowth, like that. You need to now they are thinking to develop different type of materials using via this biomedical technology research. They are synthesizing bone catalysis, muscle tones, like that. You need to develop different type of materials using the 3D bioprinter technology. And also complex microscale functional structure, micro vessels, nephrons. They are synthesizing artificial kidney also is possible. They are synthesizing initially in the nephrons. You can see the figures in right side. They are worldwide. Lot of research is going on this type of 3D bioprinting. Then how they are derived particular stem cells, and then how they use that type of stem cells. So initially, because previous and all the crop rejected, someone give the kidney to the particular person, kidney failure person, it's not possible. They will close relatives only giving the kidney. Then also they are preferring HLA typing and all. Then only they are transfer the kidneys. That is also possible. Here you can see patient derived organoid forming cells. Nowadays and all, they get stem cells from yourselves, and then they are culturing cells, stem cells, the individual organoid development. It is possible. So you have repaired any one of your heart, or anything happens in your heart, lungs, or kidney, or pancreas. It is nowadays it's possible. They are collecting your own stem cells and synthesizing particular organoid is possible. That is the feature biomedical research. Then organoid suspensions. Already told now which type of matrix, how the suspension will happen. This and all. Nowadays, bioink is available. Then they go for particular research. This kidney development and organoid development is still in research side only. If their particular type of material is available to develop the particular organ, then you easily go for organoid replacements. That is also possible. Especially you need to consider about cell density. How much cells are injected. Shear forces also important, because you are using the bio ink. So you are giving more forces, like grinding some material in mixie. Initially, you need to give small force and automatically 1, 2, 3, like that. Like that, it will be grinding and finest powder will come out. If you are pressing with cells, you are playing with cells, live cells. So something you give more shear force means automatically it will be rupture. That also become a problem. So they are consider about cell density, shear forces, remodeling matrix, that particular matrix, different type of material matrix, and then they are constructing the particular organoid. So you need to choose the cell interaction and adhesion and shear force, everything they calculated and synthesize this type of organoid model. The solid assembly and high density engineering tissue arrangement also they are analyzing. Then cell interaction they are analyzing. Finally, they transport and form the 3D organoid cell culture model. Okay, that is very, very important. Then, for example, you're going for 3D bioprinting technology, you can go for injected and extrusion, laser assisted and spheroid formation. Then you need to collect different type of stem cells. In which area you need to develop particular stem cells, you need neurons, adipose tissues, hepatocyte, muscle, this type of different type of cells you needed: fibroblast, endothelial, astrocytes, hepatocyte, smooth muscle cells, neurons, and bioink need to prepare, and then you need to go for this type of tissue, especially neural tissues, skin tissues, cardiovascular tissues, hepatic tissues, adipose tissues formation, then skeletal muscle tissue formation. Then you go for this type of organoid development. Then nowadays they are planning to introduce this type of molecular handed device for in situ bioprinting. Once they are tried with this bioprinting in a biggest manner, automatically they are trying to develop this type of bioprinting. Okay. So the bioprinting utilizes the 3D printing technology to produce the biological structure that can mimic to replace the natural tissues. Like that they are thinking. It's 2023 itself they are developed this type of molecular handed devices in situ bioprinting technology. Then another one important things in recent research is CRISPR based gene editing therapies. Nowadays, the gene editing is important tool. So you know about your whole gene, 3.5 billion genomic DNA structure, ATGC like that. Know all the gene editing technology. Already I told now, the China is developed a stem cell based type 1, type 2 diabetic reversing technology. Now, the US, America developed this type of gene editing technology to reverse the diabetic type 1, type 2 using this CRISPR-Cas9. It's possible to edit your genomics and epidemic model and enable the presence of gene in living organisms. Okay. For example, sickle cell diseases also, they are using this type of lot of cancers disease. They are now this CRISPR-Cas9 technology is the future molecular medicine for biomedical research, oncological trials, the gene editing, cystic fibrosis, cancer genes, cancer cell therapies, lot of techniques involving lot of diseases are eradicated using this molecular technology tool, CRISPR-Cas9 gene editing therapy tool. Core mechanism and schematic diagram. Usually, the Cas9 enzyme bounded to the single guided RNA, okay, which carry a region specifically designed to the recognize the complementary DNA target. First, you need to synthesize the guided RNA. Then you need to synthesize the particular targeted area. Then that it will be form the complex scRNA plus Cas9 complex located to target the particular adjacent to the PAM site. And the PAM site is 6 to 9 motif, that particular site. It's usually 5' to 3' in the particular region. Then the enzyme trigger to local DNA melting to form the RNA DNA hybrid. That is the CRISPR-Cas9 mechanism. Then cleave and repair the process. Once bounded the Cas9 two nucleus domains, then particular nucleus is in that particular domain, Cas9 domain. Leave the opposite strand of the DNA, creating the double stranded DNA break. Already you know about DNA is a double stranded structure. Then the particular enzyme go and bind that particular region and then cleave it. Then 3 to 4 days, 3 to 4 bases upstream from the PAM sequence. Then resulting double stranded break allow for the gene editing via one of the two cellular DNA repair pathway. One is non-homologic end joining and another one is homology directed repair. This two type of joining will happens using this CRISPR-Cas9 technology. So you can see the diagram, flow diagram of CRISPR-Cas9 technology. This one. First, you need to synthesize the sgRNA. Then when the sgRNA identify the particular PAM sequence, then double stranded break happens. Then it will go for either non-homologic end joining. Either you need to edit the particular gene, delete the particular gene, or insert the particular gene. Accordingly, you need to synthesize this particular HgRNA. Okay, then automatically deletion also happening and insertion also happening. Okay, this is the very important tool. Feature is no medicine. No need to take them tablet. No need to take syrup. No need to take any kind of medicine. You analyze. You have the pen drive. You can analyze whole genome sequence. You can analyze this type of gene editing technology via computer itself. Is there any gene is mutated via different sources? Automatically, you need to replace the particular gene mutation via this type of CRISPR-Cas9 gene editing technology. This is the molecular medicine important tools for future. They are trying lot of clinical trials. FDA also one of the FDA also approved this type of CRISPR-Cas9 gene editing technology. Okay, so you can see the patient derived the particular stem cells and IPs also growing. Is there anything happens? This is the process. How to the gene? This here itself you need to edit and reprogram it, and finally they can use as the medicine. Okay, gene addition and deletion is possible. Either there are still a lot of clinical trials going on mouse, and the patients also they are doing this type of CRISPR-Cas9 technology. Already the FDA approved for this type of insulin type 1 diabetic eradication via this type of gene editing technology, CRISPR-Cas9 technology. Type 1 and type 2 is eradicated. The FDA is approved for CRISPR-Cas9 gene editing technology. Okay. Then another one important. Everyone is nowadays speaking AA. What is meant by AA? Already you people also know using ChatGPT, Gemini, like that. Lot of AA is there. Each and every different type of AA is nowadays helping everyone speaking about AA. This type of how this type of AA is helping in our biomedical research. AA in personalized healthcare. How it is possible? So already you know about that smartwatch is involving to detect your everyday day activity, and then you're giving the data to the particular your data to your phone. You need to analyze how many steps are you walking, how much breath control is happening, what is your SpO2 level, what is your pressure level, what is your insulin glucose level, everything is monitored. What is your heart rate, what is your ECG, everything is possible to analyze in a smartwatch itself. So like that, they are transforming new gadgets, the bed to patient like that, bench to patients like that. So AI is very important in a healthcare industry. AI in personalized healthcare. So the role of AI in personalized medicine, first thing is genetic data analysis and interpretation. Personalized medicine, you're going for, already I told now, this CRISPR-Cas9 technology is evolved. Now they are thinking about AI in personalized medicine. So why they are going in this one? Previous slides and all you see about gene editing is possible. No need to go for medicine. Any particular medicine, drugs, paracetamol, syrup and all. You need to analyze your sequence, your data like that, DNA sequence. Automatically, then the tailored treatment plans and drugs therapies also available. They analyze any gene is small function, you go for CRISPR-Cas9, it's possible in future. Nowadays, they are thinking about any disease is available or not. They are analyzing your data, omics data, or DNA data itself, they are analyzing. Then tailored treatment plans and drug therapies. Then go for the patient satisfaction and subgroup identification. And continuous monitoring and adaptation also possible. This is the role of AI in personalized medicine. How data is analyzed? Genetic data analysis and interpretation is very important. First, the patient DNA sequence using advanced genomic techniques. If you give your blood sample, then they will extracted the DNA and then sequence your DNA and give the copy of your whole genome sequence. Then you need to analyze each and every gene. How many chromosomes in our human? 23 base pair. They are analyzing each chromosome containing how many genes. Each chromosome containing how many genes. Each genes how many base pairs. Then particular which is the transcription starting state, translation starting state, like that. All the data is available in NCBI. Each and every human 23 base pair of chromosomes is analyzed and stored your whole genome sequence data. Then any error happens, any genes modification happens, the editing is also possible via this CRISPR-Cas9 technology. So they are analyzing your particular gene sequence, then AI algorithm process the data, identifying the genetic mutation, risk factors. That is the feature. Once you collected your DNA data, then give it to the AI. Automatically, the AI is analyzing your whole genome sequence, and then is there any mutation found in any gene area, or gene restricted area, risk factor is available. Automatically, the AI analyze the particular genome sequence, then target the particular disease area, then you go for the personalized medicine. Tailored treatment plans and drug therapies also available. So the unique genetic profile interpreted by AI, clinicians design the individual treatment plans. So already the molecular medicine and genomics and proteomics is available. USA and all, they are thinking about all the disease will be cured via this type of genome editing. It's possible. That is the molecular medicine. The drug selection and dose also optimized to the. So previous and all I told now 250 milligram of paracetamol you are taken for 10 to 20 years before. Now you are taking the 650 milligram of paracetamol, $650. They are thinking about your body weight, height, and your disease profile. How much drug you need to take? It will be cure the particular area. That is also possible. The AI also nowadays helping. Those are optimized to target the specific genetic abnormalities. Then increase treatment efficacy while reducing the potential side effects. Okay. So the feature is they're reducing the side effects using this type of AI in healthcare. Okay. Then patients had subgroups and identification. The AI model, first they identify some tumor, then again and go in depth to profile the what type of tumor, which area is and which gene is disturbing, like that. Improving the risk assessment and therapy matching. Then identifying the patients who are most likely to benefit from the particular inventions. Allow for more focused clinical trials and research on that particular patients. Continuous monitoring and adaptation. So for human, itself to analyze your whole health condition. Automatically nowadays AI is helping to give the tablets. It's also possible. China is introduced a AI humanoid robot for simple diseases, fever and all. You need to stand in front of AI doctor, automatically scan everything and analyzing what type of problem you need to tell, means automatically the particular AI humanoid robot is giving the prescription, and then is there any complication happens, they suggest the particular doctor name also. It is the very important breakthrough in China. They introduced it already in market, AI in healthcare. Now we are less behind that type of work. The future, the AI doctor is arrived. The simple treatment and all, the AI doctor also helping to human being. So as a treatment progress, patient data are continuously analyzing using AI. Everyone talk about data. How they are collecting data and store in cloud. Already you know. So you have anything seen in your mobile itself, it will transfer as a data. For example, you are seeing something, you are searching for in your mobile. I need some good laptops or smartphone. Every time you are going enter into the Facebook or some other social media platform, automatically pop-up is coming. How it is possible? They are collecting your data unknowingly and sending it to the particular cloud session. They automatically pop up. This is the price for this one, this one, this one, like that. Nowadays, they are telling, the Swiggy is telling next 10 years, for whole India, I am only deciding which one eat Indian people, like that, because of this type of data collection. So already know about data collection is important for AI medicine also. Nowadays the AI and machine learning is very, very important. You need to learn this AI in healthcare. That is the future for our all biomedical student and biotechnology students also. This enable the real-time monitoring of therapeutic effectiveness, rapid detections of changes, and timely adaptation for therapy plans for new data emergencies. Okay. The personalized medicine with AI and genomics. Already told na, advanced genomic sequence combined with AI allow the therapies and diagnostic tailored to the patient genetic profile. AI-driven liquid biopsies enabling non-invasive, because doctor and all, they will inject something and then analyze and take the biopsy. Finally, this one is cancer or not, like that only doctor will decide. Nowadays AI, it deeply go monitoring that particular image itself, particular dot itself, they are analyzing without anything cutting and without anything. The AI is analyzing the biopsy inside your body and then give the what type of cancer it is, tumor profile it is. So another one technology, mRNA technology, that already the Russia is giving the mRNA vaccine for COVID-19. That's a most breakthrough, and cancer also. Russia giving the mRNA based cancer vaccine also. The personality vaccine also possible using this AI in genomic medicines. Combination comprehensive genetic profiling with AI-driven analytics and treatment. Genome sequence first. Second one is data analysis by AI, and then go for the therapy selection. How the visualization happens in the particular AI-powered liquid biopsy. Non-invasive liquid biopsy enhanced by AI detected circulating tumor DNA, RNA, or protein in the blood sample, enabling ultra diagnosis and precise the monitoring of cancer evolution using this AI-powered liquid biopsy. The schematic of blood flow while floating the genetic marker analysis by the neural network overlap. See, everything pass over itself. The AI is powered liquid biopsy is possible to analyze. Do you have a cancer or not? Like that, future medicine is coming. Then mRNA personalized vaccine, the COVID-19 pandemic and all, this type of mRNA technology is now created individually. Cancer vaccine also created Russia. The tumor mutations improving the immune targeting, and this cancer therapy also evolved. They took tissue biopsy, genomic sequence, and mRNA synthesis, vaccine, and injected. So why they took this type of biopsy? Because the AI is the personalized medicine. The generating your personalized mRNA vaccine is also possible. Generalized CRISPR-Cas9 technology also possible to cure the particular diseases, cancer-like diseases. Okay. Then multi-omics and AI, all blinds genomics with additional omics layers to reveal the complex diseases like cancer. The multiple dimensional patient profile for the precision invention. The omics also, the proteomics, omics techniques also involving and analyzing via this AI algorithms. It's also one of the feature techniques. Real-time monitoring and adaptations. So nowadays, the insulin, previous and all, they injected the insulin monitoring system. Used the insulin monitoring system, real-time monitoring system. Automatically, the insulin will be injected, like that, already available. Here also, they are monitoring real time. How they are monitoring using this type of AI? AI analyzing the sequential patient samples in a real time allowing the treatment adaptation as cancer as a disease evolving, potentially improving the outcome and minimize the side effect. Timeline showing repeated sampling, AI monitoring, and adaptive therapy branching. That is the very important visualized timeline also using this type of AI. Then therapeutic responses. This AI and machine learning predict the patient response. What is your response? Nowadays you are speaking with AI. Gemini, you need to tell this one. Gemini, you need to tell this one. Automatically it will give the solution. If you ask Gemini, I need a horror movie, or a horror story, means automatically Gemini is talking with you and giving the story also. Anything is possible. You need to speak AI, it will give the results immediately. Clinical data, assistance, clinician dose and drug selection for higher efficacy and safety. Diagram tracing and genotype to AI. Drug response prediction and outcome also possible. And self-learning healthcare. Future is self-learning healthcare is very important using this type of AI. AI model continuously learn from the expanded globally. You need to ask anything, means automatically AI is growing. Kindly you ask something, means immediately they give response. The Gemini is giving the response, and Perplexity is also giving response. Copilot also giving the response. ChatGPT also giving the response. I am very sad. You need to give some remedy, means Gemini also giving. Like that, in future is self-learning healthcare is very important, because the globally patient data is available nowadays, enhancing the treatment, personalization, and enabling more accurate risk prediction and diseases prevention strategy over the time. Then visible global of interconnected patient data sources with AI engine at the center, updating the diagnostic and the treatment protocol also possible. That is the visual feature visuals. So you can see the bioinformatics. Already I told now, you need to analyze your traditional DNA sequence using the bioinformatics workflow, ATGC using. You need to analyze your DNA data, the genetic material data, next then sequence data, then analyze the bioinformatics. Then you go for the precision medicine. Then you need to go for drug discovery. What type of disease is affected in your genome? That is the precision medicine. Then you go for which type of drug you need to take, how much mg is necessity for you, like that. Then finally you need to image the analysis the particular disease level, how it will be curious happens or not, like that only in future is going on using this AI in healthcare. Some case studies, brain tumor, especially brain cancer in a child. It's very difficult to identify. Nowadays, tumor exosome, they are identifying the tumor exon region itself exactly using this type of AI technology, AI powered. Wingless exactly identified the particular region, tumor exosome region, and then they give the therapy, treated with chemotherapy along showing the AI powered genomic enabled targeted harmful therapy, less harmful therapy, moving away from the conventional generalized treatment. They are going for this type of brain tumor studies. Okay. The predicting 3D protein structure from genetic data, that is also very important. Nearly 1,000 breast cancer cases are analyzed and then studied. The breast cancer images studied and then give. Nowadays they giving IBM Watson for oncology, giving 93% of accuracy from the data. So it will helping to identify the particular patient profile, and automatically it will generating data to the AI is generating data, and the data is giving 93% accurate result compared to the doctors. Okay. Here clinical trials and drug development, and the Microsoft also involving in this type of clinical trials and genome and clinical trials worldwide, improve the patient selection, lower the cost of 25%. Nowadays, the medicine is reduced via 25% cost via using this type of AI powered healthcare system. Then diagnostic assistance. AI powered diagnosis system measurement, already told na, AI diagnostic assistant is available in China. For example, AI has helped reduce the misdiagnosis rate and supported overworked radiologists. Sometimes they are using this type of AI helped assistance. So this is our research is focusing on developing the nanocompost, role of NRF in nanocompost, targeting inflammatory mRNAs for nanocompost, biofunctionalized nanocompost for orthopedic implant with the regenerative potential, then clinical trials and patient patents. Like that, we planned one nanocompost preparation for. And also, our research is focusing on this type of nanoparticles. Already know that nanoparticle in biomedical and healthcare is giving a lot of scaffold and matrix like structure, EVM matrix like structure, and the dactyliary systems. So you can see this type of different type of nanoparticle, fluorescence labeled nanoparticles using for tumor location. You can see the different colored image and then mixed the image, the tumor range clearly identified using this type of fluorescent nanoparticle. That is also our research. Then your university, Dr. Tirunavakaras and me also researched this type of gold nanoparticles. The gold nanoparticles synthesized from seaweeds and then analyzed for their anti-cancer activity properties. Then right now I'm doing the orthopedic implant applications. So orthopedic, why I am focusing? My mother also affected with orthopedic. My grandmother also affected with orthopedic, authorized patients. So I am focusing on this orthopedic implant applications. Applying the principle to orthopedic bone when broken down to the nanoscale is naturally nanostructure composed of collagen and hydroxyapatites. Okay. So this is the bone different length. You can see the bone has been stronger, classified outer compact layer. This is the bone structure. You need to see the bone structure. Osteocyte is there. That is the 100 to 50 nanogram. This one is collagen molecules also available. Hydroxyapatite crystal also presenting inside the bone and blood vessels also available. So these are all the structure inside the bone. So you need to recreate this type of structure. What are the different type of material they are using? That is a very, very important strategy. So using the nanotechnology in wound healing properties, I used interleukin and growth factor signalings and ECM matrix for wound healing. Alteration in the sequence led to non-healing and chronic wound healing problems and antimicrobial activities, immunomodulating antimicrobial nanoparticle, gene modification, silencing technology, and growth factor realizing nanoparticles are the different modulate available for wound care. Okay, you can see the some impingement in implant in bone replacements. This image is magnified nano engineered implant surface and it is topographically interaction with adjacent bone. There already this type of action is happening. Implant the particular different type of nanoparticle in bone replacements. So in this video, we are using 3D printed zirconium nanoparticles. Okay, you can see we are using 3D printing nanoparticle. எவ்வளவு weight add பண்ணீங்க 12 grams. இதுல வந்து நீங்க இன்னொரு இந்த இத ஸ்டாப் பண்றீங்க பேஸ்ட் அப்படியே எடுத்துட்டு you add the mouse zirconium and you come up the lens will print it. இத நான் ஸ்டாப் பண்றேன். So you can see we are synthesizing zirconium nanoparticles for bone replacements using the zirconium and polyvinyl alcohol PVA plus silver nanoparticles combinations. Why am I choosing the silver nanoparticle for this combination? The silver have already about antibacterial, anti-cancer active, antibacterial, antiviral, and antibacterial activities. So if you using this type of combined nano compost, zirconia nano compost, and replace the bone structure, zirconium is somewhat regrowthable one. So I choose this zirconium as a bone replacement. Study is almost finished, and then go for this type of different type of nanoparticle also already available in market. Our study is a little bit different. I am planning to patent that one. So synthesize initially like this, then different type of structure like this. Then I go for the particular structure. You can see the one small T25 flask. So I used a different type of cell line, epithelial cell line, MG63 cell line, adenocarcinoma cell line, and inserted our nanoparticle. I created some wounds in the particular disc, big wound like structure like wound I created, and then inserted our nanoparticles, synthesized nanocomposed particles, and then analyze is there any effect will happens inside the particular wound. So our result shows automatically our nanocompost is helping to regenerate the particular area. That is our prediction. 1 gram to 1.2 gram, this will be, you can see the difference. 1.2 gram is giving more growth formation in that particular MG63 cell line. So I made some breakthrough in that one. Just now, I filed on patent for this type of work, zirconium nanocompost, zirconium neem AG nanoparticles plus PVA compost. This is our research, nanocompost research. This one is synthesized using 3D printing technology. Then we are filing the patent for this work. And also previous work, I did some nanoparticle toxicity because I am a toxicologist and genome toxicity. We studied nanofiber, TiO2 nanofibers, structure and toxicity assessments, and genotoxicity level. We identified comet assay for nanoparticles, nanomaterials. Then this TiO2 nano cytotoxicity levels. So these are all whole summary, better understanding of clinical trials and translational biomedical research. Much progress has been made regarding the nanocompost for orthopedic implants with regenerative potential, health effects of nanoparticles, increasing the research in the health effect of clinical trials and translational biomedical research. Very, very important. So our research is focusing on orthopedic implants. So thanking you. Do you have any question you need to ask? Excuse me.
No questions, sir. Everything
No
is
questions.
clear, sir.
Okay, ma. Okay, ma. Thank you, ma. So, okay for you? Do you have any question? No.
Thank you so much, sir.
Okay, ma.
You have
Okay.
showed that video and explained us, sir. It was really interesting, sir.
Thank you, ma. Thank you.
Thank you so much, sir, for the insightful and inspiring session. Your words have surely broadened our perspectives.
So,
Now I
kind.
call up. Thank you, sir.
Thank you, ma'am, for biotechnology HOD, ma'am. Krishna, thank you for giving me these opportunities. We go for one snap.
Okay, sir.
Okay, thank you.
Video. Now, I call upon Dr. F. Fariza, ma'am, Professor and Deputy Dean HOD, Deputy HOD to give the note of appreciation.
Pleasant afternoon to one and all present here. Myself, Dr. F. Fariza, Professor and Deputy HOD, Department of Biomedical Engineering, Dr. JR Educational and Research Institute, and also the Faculty Coordinator for the Faculty Development Program on recent trends in biomedical engineering conducted from 10th November 2025 to 14th November 2025. It gives me immense pleasure to extend my heartfelt appreciation to all the students who have contributed their time, effort, and creativity towards the successful organization and smooth conduct of the 5 days FDP. Each one of you have demonstrated remarkable teamwork, dedication, and professionalism in carrying out your assigned responsibilities. I would like to appreciate the following poster designing team of students. Kutsia and Genelia from 2nd BME for designing the final poster with exceptional clarity and creativity. Renita from 2nd BME and Rubina from 3rd BME for preparing the final poster with all speaker details with great attention to detail. Again, Kutsia for designing day 1, day 4, and day 5 individual posters effectively. Rubina for designing the day 2 individual poster with elegance. Genelia for designing the day 3 individual poster thoughtfully. Event coordination and documentation team of students. Kutsia for creating the Google form for registration and for attendance, ensuring systematic record keeping. Mohita for designing the participation certificate meticulously. Rajeshwari for preparing the appreciation certificate with dedication. And Renita for preparing the event report comprehensively and on time. About the comparing and stage coordination team of students. Day 1, the comparing was done by Nithya, speaker introduction by Sri Shweta, and vote of thanks by Rubina, all from 3rd BME. On day 2, the comparing was done by Renita from 2nd BME, and speaker introduction by Rubina, vote of thanks by Sri Shweta from 3rd BME. On day 3, full comparing was done by Renita from 2nd BME, speaker introduction by Divya, and vote of thanks by Nithya from 3rd BME. On day 4, comparing was done by Shweta, speaker introduction by Rubino of 3rd BME, and vote of thanks by Renita of 2nd BME. On Friday, the comparing speaker introduction both was done by Rubina. And special acknowledgement goes to Parthasarathi, Secretary, NBC, for serving as the main source of coordination and communication among all the student committee for preparation of various event-related details and for his consistent support throughout the planning and execution stage of this FDP. And a special appreciation and acknowledgement goes to Rubina of 3rd BME, Joint Secretary NBC, and Renita of 2nd BME, Executive Member of NBC, for efficiently managing the meeting links and ensuring the smooth online conduction of the event across all the 5 days. From the student side, all the three have supported our team of faculty with almost most part of the FDP. Your collective efforts have made this event a grand success. The commitment, creativity, and collaboration displayed by each one of you truly reflects the spirit of teamwork and excellence that our department stands for. Sincere appreciation and best wishes for all your continued success, and we expect the same commitment, creativity, and collaboration in the future also. Thank you, each and every student who had contributed greatly for this FDP successful completion. Thank you.
Thank you so much, ma'am. Thank you for
Thank you, ma.
expressing. Thank you so much, sir. To formally express our gratitude to all who made this event possible, now I call upon B.R. Shankari, ma'am, Assistant Professor, to deliver the vote of thanks. Over to you, ma'am.
Good afternoon to one and all present here. It's my great privilege to deliver the vote of thanks on behalf of Department of Biomedical Engineering for today's successful session of the 5 days virtual faculty development program on recent trends in biomedical engineering, organized in association with the Academic Staff College and Next Gen Biomedical Committee (NBC). First and foremost, I would like to express my heartfelt gratitude to our respected HOD Madam, Dr. K.S. Divya, for providing us the wonderful opportunity and continuous support in organizing the FTP. My sincere thanks to our distinguished chief guest, Dr. P. Cynthia Ma'am, Dr. Sendil Kumar, Dr. Ram Kumar, Mr. Vinod Kumar, and Dr. P. Leelavati for taking time for their busy schedule to share their valuable insights and knowledge with us. I would also like to extend my gratitude to all of our faculty members for their support and coordination in organizing this FTP smoothly and successfully. A special thanks to our Honorable President Sir and the management for their constant engagement, motivation, and guidance in conducting such meaningful academic programs. Last but not least, I thank all the participants for their active participation and enthusiastic participation, which made this event a great success. Thank you, one and all. Thank you.
Thank you all. Have a wonderful day. The feedback link and the attendance link will be posted.
Thank you, ma'am.